हेलो फ्रेंड्स, वेलकम बैक टू माय चैनल एकेडमी ऑफ सेंशिया। और आज हम देखने वाले हैं रिटन नोट्स फॉर बीएससी एंड एमएससी। तो मैं आपको ये नोट्स सिर्फ रीड करके बताऊंगी और आप लोग इसे रिटर्न कर सकते हैं या फिर पढ़ाई भी कर सकते हैं या जूम करके स्क्रीनशॉट लेके भी नोट्स अपने बना सकते हैं। तो जो हमारा फर्स्ट पॉइंट है वो है असेसमेंट ऑफ स्पर्म फंक्शन। सो the first paragraph is the main sperm function is to reach the ovum and fuse with to deliver to subcellular extracts in the male pronucleus that contains the genetic material. The centrioles that are structure that help organize the microtubule cytoskeleton. Next जो main heading है वो है anatomy. Anatomy means anatomy. The mammalian sperm cell consists of head, neck, and middle piece. Contains the nucleus with densely coiled chromatin fibers surrounded entirely by an acrosome which contains enzyme used for penetrating the female egg. The neck contains the sperm centriole. The midpiece has a central filament are with many mitochondria spiraled around it, used for ATP production for the journey through the female cervix, uterus and uterine tubules. See on the next page. The tail or a flagellum acts as the lashing movement of the propel the spermatocyte. During the fertilization, sperm provide the essential parts to the oocyte, a single or activating factor which causes the metabolically dormant oocyte to activate the haploid paternal genome. The centriole, which is responsible for forming the centrosomes and microtubule system. जो नेक्स्ट पॉइंट है हमारा वो है लास्ट वन और सेकंड वन पॉइंट इज द असेसमेंट ऑफ ओरिजिन एज अ फंक्शन. सो द स्पर्मेटोजोआ ऑफ एनिमल्स are the produced through the spermatogenesis inside the male gonads testicles via mitotic division. The initial spermatozoon process takes around to 2 days to complete. 70 days to complete. नेक्स्ट पेज अगर हम देखेंगे सो यहाँ पे पॉइंट देख सकते हैं आप. द स्पर्मेटेड स्टेज इज वेयर द स्पर्म डेवलप द फैमिलियर टेल मैच्योर. द नेक्स्ट स्टेज वेयर इट becomes fully mature takes around 60 days when it is called a spermatozoon. Sperm cells are carried out of the male body in a fluid known as the semen. Human sperm cells can survive within the female reproductive tract for more than 5 days post-coitus. Semen is produced in the seminal vesicles, prostate gland and urethral gland. नेक्स्ट पॉइंट हम लोग देख सकते हैं असेसमेंट ऑफ स्पर्म क्वालिटी. सो असेसमेंट ऑफ स्पर्म क्वालिटी, स्पर्म क्वांटिटी एंड क्वालिटी आर the main parameters in semen quality, which is a measure of the ability of a semen to accomplish fertilization. Thus in humans it is a measure of fertility. In a man, the genetic quality of a sperm as well as its volume and motility all typically decreases with age. नेक्स्ट पेज देखते हैं हम. DNA damages present in sperm cells in the apparatus after meiosis but before fertilization may be repaired in the fertilized egg, but if not repaired can have a serious deleterious effects on fertility and the developing embryo. Human sperm cells are particularly vulnerable to free radical attack and the generation of oxidative DNA damage. नेक्स्ट पॉइंट जो है वो है हमारा assessment of sperm size. So related to sperm quality in sperm size, at least some animals had some sperms of a fruit fly. Drosophila are made up of 5.8 centimeter long, about 20 times as long as the fly itself. Longer sperm cells are better than their shorter counterparts of displacing competitors from the female seminal receptacles. नेक्स्ट पॉइंट जो है हमारा वो है द स्पर्मेटेड स्टेज इज वेयर द स्पर्म डेवलप्स द फैमिलियर टेल. द नेक्स्ट स्टेज वेयर इट बिकम फुल्ली मैच्योर टेक्स अराउंड 60 days when it is called as a spermatozoon. Sperm cells are carried out of the male body in a fluid known as a semen. Human sperm cells and survive within the female reproductive tract or for more than 5 days post-coitus. नेक्स्ट पॉइंट जो है हमारा, द बेनिफिट टू फीमेल इज दैट ओनली हेल्थी मेल्स कैरी गुड जीन्स दैट कैन प्रोड्यूस लार्ज स्पर्म इन अ सफिशिएंट क्वांटिटीज टू आउटकम्स देयर कॉम्पिटिटर्स. And this point is a assessment of sperm size. So sperm cells are carried out of the male body in a fluid known as a semen. The female reproductive tract or more than 5 days post-coitus. Semen is produced in the seminal vesicles, prostate gland and urethral gland. Next point is the assessment of sperm qualities. Sperm quantity and quality are the main parameters in quality, which is a measures of the ability of a semen to accomplish fertilization. Thus in human it is a measure of fertility in a man. The genetic quality of sperm as well as its volume and motility are typically decreases with age. DNA damage is present in sperm cells in the period after meiosis, but before fertilization may be repeated in the fertilized egg, but it not repaired can fertilized egg, but it not repaired can have a serious deleterious effects on a fertility and the developing embryo. Human sperm cells are particularly vulnerable to take a free radical and the generation of oxidative DNA damage. Assessment of sperm size related to sperm quality in sperm size, at least in some animals. For instance, the sperm of some animals or some sperm of a long about 20 times as long as the fly itself. So longer sperm cells are better than their shorter counterparts of displacing competitors from the female seminal receptacles. The benefit to female is that only healthy males carry good genes that can produce large sperm in a sufficient quantity to outcome their competitors.